For Unit 1, Chemistry of Life, for AP Biology, we're going to be covering Section 1.5, Structure and Function of Biological Macromolecules. Just as a disclaimer, AP Biology is a registered trademark of College Board and is not involved in the development of the products we're going to be talking about. Now, when you're covering the overall view of what you need to understand for Unit 1, this table really covers the macromolecules, the structure, the linkage bond, the types, the functions, and examples. So not only does it cover section 1.5, I should say, but it also covers section 1.4 and 1.3 and 1.6. So if you're looking for 1.3 onward and you're looking for a brief understanding of everything that you need to know related to macromolecules, this table does it for covering that topic. Now, in overview, we have four major types of macromolecules: carbs, lipids, proteins, and nucleic acids. For definitions and for free response questions, you are expected to understand the similarities and differences between the four groups. Please also understand in later units you go more into detail about nucleic acids. So that's something that you need to know more in detail about, and we'll talk about the differences in that. Now, the reason why macromolecules are important and understanding the chemical compound associated with it is because we understand that functionality, or what the purpose of the macromolecules are, is really determined by the structure itself. So we have subunits, which are monomers, which build upon each other to form a macromolecule. And depending on what monomer you do have, you really are changing the overall functionality of that macromolecule. So first we're going to cover carbs, and that is the first macromolecule. You need to understand that there are basic structural understandings and bondage that we'll cover in just a second that we're focusing on for this specific topic. So when we talk about carbs, you see the picture on the right here, and you'll notice the commonality of the ring structure and or the chain structure that you see here, and three main elements, which is carbon, and then hydrogen, and then oxygen, which are the three main elements that are formed in carbohydrates. For monosaccharides, you're talking about single sugar units. So you either see one ring or you see one long chain, and that is your monosaccharides. Now, when we're talking about disaccharides, we're talking about having those monosaccharides conjoined together by this linkage, the glycosidic linkage. And so if you see two of them joined by one link, that is considered a disaccharide, and di meaning two. So that's two sugar units, two, or I should say, if you have more, then we are falling under the category of polysaccharides. So those have the multitude of chains, more than two chains together of those monosaccharides, so long chains. And when we think about carbohydrates, we're thinking about things like breads. So, for example, in social media, you might say, oh my gosh, this bread has a lot of carb, which is true. And the reason why is because it's a major energy source, and it provides the nutritional element of energy that you need to really focus on what is associated with carbs. And sometimes you're talking about structure as well. So a cell itself on the outside might have a carb that is located there to hone in for structural support. Now let's talk about lipids, which are oftentimes composed of carbon, hydrogen, and oxygen, and sometimes phosphorus. They're composed of fats, oils, and phospholipids, and they commonly are functioned in energy storage, cell membrane, and structure. In the picture seen here, we talk about lipids as the third row on the right here. So when we're talking about lipids, we're talking about having those ester bonds. And I want to point out the difference between saturated and unsaturated fats, which are a type of lipid. So as noted earlier, lipids composed of highly fats, oils, steroids, phospholipids. And the key distinguishing factor is to understand the difference between saturated and unsaturated fats. Saturated fats tend to have single carbon bonds, so they're distinguished with that C chain, but only having a single bond between represented by one line, and they're often solid at room temperature. I want you to think of butters. Unsaturated fats, however, are double carbon-carbon bonds, so they're going to have that two-line distinguishing factor, and that usually means that they're liquid at room temperature. So I want you to think of things like oils that are seen on the page here. Now, when we talk about the other category, we're talking about proteins. And proteins are unique because when we talk about proteins, we're talking about distinguishing and understanding that there are different levels and that they have the unique property of nitrogen that are involved. Think amino or aminoids. Now, proteins are bonded with peptide bonds. They have monomers that are amino acids, and there are 20 different types of amino acids. They can combine together, be a dipeptide if there are two, and if many, then it's called a polypeptide if there's a long chain of amino acids. So when we talk more in detail about the different structures, primary, secondary, tertiary, and quaternary, I want you to understand the difference and key features between the four different groups. Primary is the most basic. We're just talking about amino acids just connecting together. However, once we're talking about whether or not it's flat or it's on a tailwind, meaning there's some sort of bend, we're trying to distinguish between whether or not it's alpha or beta. So alpha has this sort of twist-like shape associated with it, and then beta is more flat. So that's the difference in the secondary structure is we're trying to distinguish whether or not it's flat or whether or not there's a twist on itself. Once it has some sort of depth through it, you want to think of the term 3D. So 3D has the term three, or tertiary also means three, and that's what tertiary means. It means that we have some sort of folding, which leads to some sort of 3D structure to it, as shown here, where you have a bunch of either alpha and beta or a combination of both to make this 3D structure associated with the protein. And lastly, you can have quaternary proteins, which is this idea of the multiple polypeptide chains together. Now, with proteins, I want you to think of the most common type of protein, which are enzymes. So enzymes come up a lot in biology, and that's because they have very unique characteristics and they're very important for the functionality of the human body. They result in a loss of biological activity oftentimes, and enzymes in general fluctuate based on the behavior related to heat or some sort of temperature, pH change, or chemical change. So temperature or pH or chemistry related to proteins can often alter the way that the protein reacts. When we talk about the final group of macromolecules, we're talking about nucleic acids, which basically is our genetic information. They hold our blueprint and are grouped into two main categories, DNA and RNA. While the specifics are more detailed in further units, it is favorable to understand that nucleic acids are covered in two main giant categories, that DNA category and RNA category. Now, when we talk about nucleic acids, as stated before, we're talking about DNA and RNA. And DNA and RNA have their own unique differences as well. So, for example, DNA is double-stranded, so you'll see these two strands here, while RNA is one-stranded, so you'll see that one strand there. This sugar is known as deoxyribose for DNA, which is why it's called DNA, and RNA has ribose, which is why it's called ribonucleic acid. Once again, they carry the genetic information, and the basic backbone of what nucleic acids are nitrogenous bases, sugars, and phosphate groups. So the way that you can forego or understand DNA structure, which is further detailed in further units, is understanding what the backbone is and the parts. So when we talk about DNA, we're talking about a phosphate backbone, and that's that pink color right here. We have these letters, and they're called nitrogenous bases. For DNA, you have A, T, C, and G. And what we do know is that A likes to bind with T and C likes to bind with G. Now, with that pairing, we also know that the bonds between those nitrogenous bases are hydrogen bonds. And we call them anti-parallel because it's the way the direction of the strands are. So, for example, they go in one direction this way while the other strand goes in the other direction this way. So just to recap, you have A binding to T always and C binding to G always for DNA. And they are bonded together with hydrogen bonds and have a phosphate background. RNA is very similar. However, it is noted that the nitrogenous bases are different for thymine. There is no thymine for RNA. Instead, it has letter U or uracil in its replacement. So we know also that RNA is single-stranded, and the types of RNA include mRNA, tRNA, and rRNA. And the proper functionality is protein synthesis. So now it's quiz time. So we're going to cover some questions. Take a moment and read the following questions shown here. Make sure to pause your screen and read the following question. Once completed, let's unpause and review the question together. Okay, now that you've completed reading the question, you can see here that they're talking about genetic information. The main category that is involved in genetic information is nucleic acids. So the answer would be D. Remember, nucleic acids are subcategorized into DNA and RNA. Mostly DNA is our genetic makeup. So that's the information that they're trying to present in this question. Let's take a look at this question here. Take a moment, pause your screen, and review the question. Once you're done selecting your answer, make sure to unpause the video. Okay, now that you're done, reviewing the question. The question asks, the primary structure of a protein is determined by which of the following? And the answer is amino acids. Remember, amino acids are the basic building block of any protein. And so if you remember, there's primary, secondary, tertiary, and quaternary. And quaternary is known for having multiple polypeptide chains. So quaternary would be letter D. If we're talking about the understanding of the folds, that would be understanding more of the 3D structure, I should say. That would be tertiary. If it's alpha, beta, that's secondary. And if we're talking about a basic sequence, we're talking about primary. So the correct answer in this case is answer choice A. I hope that helps understand a little bit more about macromolecules. This is the very unique foundations of AP biology or college biology. Make sure to hit that subscribe button for more videos related to this type of topic. Hey, you want to know a little secret? If you hit the subscribe button, you'll be able to get up-to-date information related to AP Biology and the digital SAT math test. Making sure that you're able to understand the content is just the first step. So if you want updated additional information, make sure to click the link tree in our bio. We hope that you subscribe because each new subscriber gives us motivation to create more up-to-date content for you, students, parents, and teachers. Enjoy!